हेलो एवरीवन, वेलकम टू NCERT ऑडियोबुक। आज हम क्लास 12th का चैप्टर 6 को रीड करेंगे। उसका नाम है मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस। In the previous chapter, you have learned the inheritance patterns and the genetic basis for such pattern. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is the genetic material. At least for the majority of the organism, in class 11th, you have learned that nucleic acid are polymers of nucleotides. Deoxyribonucleic acid DNA and ribonucleic acid RNA are the two types of the nucleic acid found in living system. DNA acts as a genetic material in most of the organisms. RNA through it also acts as a genetic material in some viruses. Mostly functions as a messenger. RNA has an additional role as well. It functions as adapter, structural, and in some cases as a catalytic molecule. In class 11, you have already learned the structure of the nucleotides and the ways these monomers units are linked to form the nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA and DNA, transcription, the genetic code that determines the sequences of amino acids in protein, the process of protein synthesis, translation, and elementary basis of their regulation, the determination of complete nucleotide sequence of a human genome during last decade has set in the new era of genomics. In the last section, the essential of the human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In the subsequent section, we will understand that why it is the most abundant genetic material and what its relationship with RNA. 6.1 The DNA. DNA is a long polymer of deoxyribonucleotides. The length of the DNA is usually defined as the number of the nucleotides or a pair of the nucleotides referred to the base pairs represent in it. This also is the characteristics of an organism. For example, a bacteriophage known as theta into 174 has 5386 nucleotide. Bacteriophage lambda has 48,502 base pairs. Escherichia coli has 4.6 into 10 to the power 6 base pair, and the haploid content of the human DNA is 3.3 into 10 to the power 9 base pair. Let us discuss the structure of such a long polymer. Structure of a polynucleotide chain. Let us recapitulate the chemical structure of the polynucleotide chain, DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA, and deoxyribose for DNA, and the phosphate group. There are two types of the nitrogenous bases: purines, adenine and guanine, and pyrimidines, cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA, and thymine is present as a DNA. Uracil is present in RNA at the place of thymine. A nitrogenous base is linked to the pentose sugar through a N-glycosidic linkage to form the nucleoside. Such adenosine or deoxyadenosines, guanosine or deoxygynosine, cytidine and deoxycytidine, and uridine and deoxythymidine. When a phosphate group is linked to the 5' end OH of the nucleoside through the phosphodiester linkage, A corresponding nucleotide or deoxyribonucleotide, depending upon the type of the sugar present, is formed. Two nucleotides are linked through 3' to 5' phosphodiester linkage to form the dinucleotide. More nucleotide can be joined in such a manner to form the polynucleotide chain. A polymer thus formed has at one end and the free phosphate moiety at 5' end of the ribose sugar, which is referred to the 5' end of the polynucleotide chain. Similarly, at the other end of the polymer, the ribose has a free chain. Similarly, at the other end of the polymer, the ribose has free 3' OH group, which is referred to 3' end of the polynucleotide chain. The backbone in the polynucleotide RNA world. From foreign discussion, an immediate question becomes evident: which is the first genetic material? It shall be discussed in detail in the chapter of chemical evolution, but briefly we shall highlight some of the facts and points. RNA was first genetic material. There is now enough evidence to suggest that essential ripe processes such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as the genetic material as well as the catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and are not by protein enzymes. But RNA being the catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that makes it more stable. DNA being double-stranded and having complementary strand further resist change by the evolving a process of repair replication. While proposing the double helical structure of DNA, Watson and Crick had immediately proposed a scheme for replication of DNA to quote their original statement, that is as follows: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick in 1953. The scheme suggested that the two strands would separate and acts as a template for the synthesis of new complementary strands. After the completion of the replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as the semi-conservative DNA replication. The experimental proof. It is now proven that the DNA replicates semi-conservatively. It was shown first in the Escherichia coli and subsequently in higher organisms such as plants and human cells. Matthew Meslon and the Franklin Sahil performed the follows experiment in 1958. They grew E. coli in the medium containing an NH415Cl isotope of nitrogen as only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. These heavy DNA molecules could be distinguished from the normal DNA by centrifugation in the cesium chloride (CaCl) destiny gradient. Please note the N15 is not a radioactive isotope and it can be separated from N14 only based on the densities. Then they transferred the cells into the medium with the normal NH4Cl14 and took samples at various definite time interval as the cells multiplied. They extracted of the DNA that remained as double-stranded helix. The various samples were separated independently on CaCl gradient to measure the densities of DNA. Can you recall what centrifugal force is and think why a molecule with higher mass density would sediment faster? Thus, the DNA that was extracted from the culture one generation after the transfer of the N15 to N14 medium, that is after 20 minutes, E. coli divides in 20 minutes, had a hybrid of the immediate densities. DNA extracted from the culture after another generation, that is after 40 minutes, second generation was composed is equal to the amount of this hybrid DNA and light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportion of the light and hybrid densities DNA molecule? Very similar experiment involving use of the radioactive thymidine and detect the distribution of the newly synthesized DNA in the chromosome was performed on Vicia faba by Tyler and colleagues in 1958. The experiment proved that the DNA in chromosomes also replicates semi-conservatively. The machinery of the and the enzymes. In the living cells such as E. coli, the process of the replication requires a set of the catalysts or enzymes. The main enzymes is referred to the DNA-dependent DNA polymerase. Since it uses the DNA template to catalyze the polymerization and deoxynucleotides, these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of the nucleotides in a very short time. E. coli that has only 4.6 into 10 to the power 6 base pair, it with human whose diploid contains 6.6 into 10 to the power 9 base pair, completes the process of replication within 18 minutes. That means the average rate of the polymerization has to be the approximately 2000 base pair per second. Not only do these polymerize have a be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during the replications would result into mutation. Therefore, furthermore, energetically replication is a very expensive process. Deoxyribonucleotides triphosphatase serve several dual purposes in the addition to acting the substrate. They provide energy for polymerization reaction. The two-terminal phosphate in the deoxynucleotides triphosphatase are high energy phosphate, same in case of the ATP. In addition to the DNA-dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For the long DNA molecule, since the two strands of the DNA cannot be separated in its entire length due to the very high energy requirement, the replication occurs within the small opening of the DNA helix, referred to as the replication fork. The DNA-dependent DNA polymerase catalyzed polymerization only in the one direction, that is 5' to 3'. This creates some additional complications at the replicating forks consequently. On one strand, the template with the polarity is 3' to 5', the replication in continuous, while on the other, the template with the polarity 5' to 3'. It is discontinuous. The discontinuously synthesized fragments of the larger latter joined by the enzyme DNA ligase. The DNA polymerase on their own cannot initiate the process of the replication. Also, the replication does not initiate randomly at any place in the DNA. There is the definition region in the E. coli DNA where the replication originates. Such regions are termed as the origin of replication. It is because of the requirement of the origin of the replication that a piece of the DNA, if needed to be propagates during the recombinant DNA producers, requires a vector. The vectors provide the origin of the replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the sex cycle. The replication of DNA and the cell division cycle should be highly coordinated. A failure into the polyploidia, you will learn the detailed nature of the origin and the processes occurring at this site in higher classes. Transcription. The process of copying genetic material information from one strand of the DNA into RNA is termed as the transcription. Here also, the principle of the complementary govern the process of the transcription. except the adenosine now forms the base pair with the uracil instead of the thymine. However, unlike in the process of replication, which once set in the total DNA of an organism gets duplicated, in transcription only a segment of the DNA and only one of the strands is copied into RNA. This necessitates defining the boundaries that would demarcate the region and the strands of the DNA that would be transcribed. Why both the strands are not copied during the transcription has the simple answer. First, if both the strands act as a template, they would code for RNA molecule with different sequences. Remember, the complementary does not mean identical, and in turn, if they codes for proteins and the sequence of the amino acids in the proteins would be different. Hence, the one segment of the DNA would be coding for the two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other. Hence, would form a double-stranded RNA. This would prevent RNA from being translated into the protein, and the exercise of transcription would become to fertile one. Transcribed unit. The transcription unit in the DNA is defined primarily by the three regions in the DNA: a promoter, a structural gene, and a terminator. There is a convention of the defining the two strands of the DNA in the structural gene of the transcription unit. Since the two strands have opposite polarity and the DNA-dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5' to 3'. The strands that has polarity 3' to 5' acts as a template and is also referred to as the template strand. The other strand which has the polarity 5' to 3' and the sequence same as RNA, except the thymine at the place of the uracil, is displaced during the transcription. Strangely, the strands which does not codes for anything is referred to as a coding strand. All the references point while defining a transcription unit is made with the coding strand. To explain the point, a hypothetical sequence from the transcription unit is represented below. This is the template strand and coding strand. The promoter and the terminator flank of the structural gene in a transcription unit. The promoter is said to be located towards the 5' end upstream of the structural gene and reference to the made of with the respect of the polarity of the coding strand. It is the DNA sequence that provides binding site for the RNA polymerase, and it is the presence of the promoter in the transcription unit that also defines the template and coding strands. By switching this position with terminator, the definitions of coding and template strand could be reversed. The terminator is located towards the 3' end downstream of the coding strand, and it usually defines the end of the process of the transcription. There are additional regulatory sequences that may be present further upstreams and downstreams to be the promoter. Some of the properties of these sequences shall be discussed while dealing with the regulations of gene expression. Transcription unit and the gene. A gene is defined as the functional unit of the inheritance through there is no ambiguity that the genes are located on the DNA. It is difficult to literally define a gene terms of a DNA sequences. The DNA sequences coding for the tRNA or RNA molecule also defined a gene. However, by defining the cistron, a segment of the DNA coding for the polypeptide, the structural gene in the transcription unit could be said to be the monocistronic, mostly in eukaryotic, or polycistronic, mostly in bacteria or prokaryotic. In eukaryotes, the monocistronic structural gene have interrupted coding sequences, and the genes of the eukaryotics are splits. The coding sequences are expressed sequence are defined as exons. Exons are said to be those sequences that appears in the mature and processed RNA. Exons are interrupted by the introns. Introns or intervening sequence do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of the gene in terms of the DNA segment. Inheritance of the characters also affected by the promoters and regulatory sequences of a structural genes. Hence, sometimes the regulatory sequences are loosely defined as the regulatory genes, even though these sequences do not code for any RNA or protein. Types of the RNA and the process of transcription. In bacteria, there are three major types of RNAs: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. The mRNA provides the template, tRNA brings amino acid and reads the genetic code, and rRNA plays the structural and catalytic role during the translation. There is a single DNA dependent RNA polymerase that catalyzes transcription of the all types of the RNA in bacteria. RNA polymerase binds to the promoter and initiates the transcription. Initiation, it is uses the nucleoside triphosphate as a substrate, and polymerase in the template dependent fashion following the rules of the complementarity. It somehow also facilitates opening the helix of the continuous elongation. Only a short stretch of RNA remains bound to the enzymes. Once the polymerase reaches the terminator region, the nascent RNA falls off. So also the RNA polymerase. This results in the terminations of the transcription. And intriguing questions is that how is the RNA polymerase able to catalyze the three steps, which are initiation, elongation, and the termination. The RNA polymerase is only capable of catalyzing the process of elongation. It associated transiently with the initiation factor and termination factor. To initiate the terminate and the transcription respectively, association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. In bacteria, since the mRNA does not require any processing to become the active, and also since transcription and translation takes place in the same compartment, there is no separation or capsule of the nucleus in bacteria. Many times translation can begin much before in the mRNA is fully transcribed. Consequently, the transcription and the translation can be coupled in bacteria. In eukaryotics, there are two additional complexities. There are at least three RNA polymerase in the nucleus in addition to the RNA polymerase found in organelles. There is a clear-cut division of the labor. The RNA polymerase 1 transcribed rRNAs 28S, 18S, and 5.8S, whereas the RNA polymerase 3 is responsible for the transcription of tRNA, 5S RNA, and snRNAs, small nuclear RNAs. The RNA polymerase 2 transcribes the precursor of the mRNA and the heterogeneous nuclear RNA or hnRNA. The second complexity is that the primary transcript contain both the exon and the introns are non-functional. Hence, it is subjected to the process called splicing, where the introns are removed and exons are joined in the definite order. hnRNA undergoes the additional processing called the capping and tailing. In capping, an unusual nucleotide methyl gynosine triphosphate is added to the 5' end of the hnRNA. In telling, the adenylate residue 200 to 300 are added in the 3' end of the template independent manner. It is fully processed hnRNA, now called mRNA, that is transported out to the nucleus for the translation. The significance of such a complexity is how beginning to be the understood the spliced genes arrangements represent probably on the ancient features of the genome. The presence of the introns is reminiscent of the antiquate, that is the process of splicing represent the dominance of the RNA world. In the recent time, the understanding of RNA and RNA-dependent processes in the living system have assumed more importance. Genetic code. During the replication and transcription, a nucleic acid was copied to the form the another nucleic acid. Hence, these processes are also easy to conceptive on the basis of complementary. The process of translation requires the transfer of the genetic information from polymer to nucleotide to the polymer for amino acid. Neither does any complementary exist between the nucleotides and the amino acid, nor could any be drawn theoretically. There existed ample evidence through a support of the notion that change in the nucleic acid genetic material were the responsible for the change in the amino acid in protein. These lead to be the proportions of the genetic code that could be direct the sequence of the amino acid during the synthesis of the protein. If determining the biochemical natures of the genetic material and the structure of the DNA was very exciting, the proportion and the deciphering of the genetic code were most challenging in the very true sense. It requires involvement of the scientist for several disciplines: physiologist, organic chemist, biochemist, and genetics. It was the Gregor Gamow and the physicist who argued that since there are only 4 bases, and if they have to code the 20 amino acid, the codes should be constitute the combinations of bases. He suggested that in order to code for all the 20 amino acid, the code should be made up of 3 nucleotides. This was a very bold proposition because the premature combinations of 4 is to power 3, 4 into 4 into 4 would generate 64 codons, generating many more codons than required. Providing proof that the codons was triplet was a more daunting task, and chemical method developed by the Har Gobind Khorana was instrumental in the synthesizing RNA molecules with defined combinations of bases. Marshall and Nigrin's cells-free system of the protein synthesis finally helped the codes to be described in the polymerase RNA with the defined sequences in the template-independent manner. The silent features of the genetic code are as follows. The codon is triplet. 61 codons codes for the amino acid, and the 3 codons do not code for any amino acid, hence they are function as stop codons. One codon codes only one amino acid, hence it is the unaminogeous and specific. Some amino acids are coded by more than one codon, hence they are coded degenerates. The codons is read in mRNA is a contiguous fashion. There are no punctuations. The codes is nearly universal. For example, the bacteria to human UUU would code for the phenylamine. Some exceptions to these rules have been found in mitochondrial codons and in some protozoans. AUG has the dual function. It codes the methionine and it also acts as the initiator codon. It follows the sequences of nucleotide in mRNA, predict the sequence of the amino acid coded by it, text in the checkerboard, UAG and like this. Now try to opposite the following the sequence of the amino acid coded by an mRNA, predict the nucleotide sequence in the RNA. Met, fet, fec, 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 fec. Do you face any difficulty to predicting the opposite? Can you now correlate which two properties of the genetic code you have to learn? Is formed due to the sugar and phosphate. The nitrogenous bases linked to the sugar moiety project from the backbone. In RNA, every nucleotide received has an additional OH group present at second position in the ribose. Also in RNA, the uracil is found at the place of thymine. 5-methyl uracil, another chemical name for thymine. DNA as the acidic substance present in the nucleus was first identified Friedrich Mescher in 1869. He named it the nuclein. However, due to the technical limitations in isolating such a long polymer intact, the elucidations of the structures of the DNA remains elusive for the very long period of time. It was only in 1953 that James Watson and the Friedrich Crick, based on the X-ray diffraction data produced by the Mercury, Marie Wilhelm and the Rosalind Franklin proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks of their propositions was based pairing between the two strands of the polynucleotide chain. However, this proposition was also based on the observation of the Erwin Chargaff that for the double-stranded DNA, the ratio between the adenine and thymine and guanine and cytosine are constant and equal 1. The base pairing confers a very unique property to the polynucleotide chain. They are said to be complementary to each other, and therefore if the sequence of the bases in one strand is known as the sequence of the other strand can be predicted. Also, if each strand of the DNA, let us call it as a parental DNA, acts as a template for synthesis of the new strands, the two double-stranded DNA, let us call them the daughter DNA, thus produced, would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA become very clear. The silent features of the double helix structure of DNA are as follows. It is made up of two polynucleotide chains where the backbone is constituted by sugar phosphate and the base project inside. The two chains have an anti-parallel polarity. It means if one chain has a polarity 5' to 3' and the other has 3' to 5', the bases in two strands are paired through hydrogen bond forming the base pairs. Adenine forms two hydrogen bonds with the thymine forms opposite strand and vice versa. Similarly, guanine is bonded with the cytosine with three hydrogen bonds. As a result, always a purine comes opposite to pyrimidines. This generates the approximately uniform distance between the two strands of the helix. The two chains are coiled in the right-handed fashion and the pitch of the helix is 3.4 nanometer and there are roughly 10 base pair in each turn consequently. The distance between the BP in the helix is approximately equal to 0.34 nanometer. The planes of the one base pair stacks over the other is double helix. This in addition to the H bonds confirms stability of the helical structure. Compare the structure of the purines and pyrimidines. Can you find out why the distance between the two polynucleotide chain in the DNA remains almost constant? The propositions of the double helix structures for the DNA and its simplicity in explaining the genetic implications become revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA to protein, from DNA transcription to form mRNA, and from mRNA translation to form protein. That complete process we called central dogma. In some of viruses, the flow of the information is reverse direction, that is from RNA to DNA. Can you suggest a simple name for the process? Packaging of DNA helix. Taken the distance between the two consecutive base pairs as a 0.34 nanometer, if the length of the DNA double helix in the typical mammalian cell is calculated simply by multiplying the total number of the base pair with distance between the two consecutive base pair. That is the 6.6 into 10 to the power 9 base pair into 0.34 into 10 to the power minus 9 base pair. It comes out to be approximately 2.2 meters, a length that is far greater than the dimensions of the typical nucleus. How is such a long polymer packed in a cell? If the length of the E. coli DNA is 1.36 mm, can you calculate the number of the base pair in E. coli? In prokaryotics such as E. coli, Though they do not have a definite nucleus, the DNA is not scattered throughout the cell. DNA being negatively charged and healed with some proteins that have the positive charge and region termed as a nucleoid. The DNA is a nucleoid and organized a large loop held by the proteins. In eukaryotic, this organization is much more complex, and there is a set of the positively charged basic proteins called histones. A protein acquires charge residue with the charged aside chains. Histones are rich in the basic amino acid residue lysine and arginines. Both amino acid residues carry positive charge in their side chains. Histones are organized to form the unit 8 molecules called histone octamer. The negatively charged DNA is wrapped around positively charged histone octamer to form the structure called nucleosome. A typical nucleosome contains 200 base pair of the DNA helix. Nucleosome constitute the repeating units of the structure of nucleus is called chromatin. Thread-like strains bodies seen in the nucleus. The nucleosomes in the chromatin are seen as beads on string. Structure when viewed under the electron microscope. Theoretically, how many such beads do you imagine are represent in the mammalian cell? The beads on the string structure in the chromatin is packed to form the chromatin fibers that are further coiled and condensed at a metaphase stage of the cell division to form the chromosome. The packaging of the chromatin at higher level requires additional set of the proteins that collectively are referred to as non-histone chromosomal protein. In a typical nucleus, some of the region of the chromatins are loosely packed and are referred to the euchromatin. The chromatin that is more densely packed and stained dark as called the heterochromatin. Euchromatin are said to be the transcriptionally active chromatin, whereas the heterochromatin is inactive. The search for the genetic material. Even through the discovery of the nucleon by the Mischur and the propositions of the principles of the inheritance by the Mendels were almost at the same time, but that the DNA acts as the genetic material took long to be discovered and proven. By 1926, the quest of the determine the mechanism for the genetic inheritance has reached the molecular level. Previous discoveries by the Gregor Mendel, Walter Sutton and Thomas Hunt Morgan and numerous other scientists had narrowed the search of the chromosomes located in the nucleus of the most cells. But the question of what molecule was actually the genetic material had not been answered. Transforming principle. In 1928, Frederick Griffith, in the series of experiments with Streptococcus pneumonia, bacterium responsible for pneumonia, witnessed a macrolus transformation in the bacteria. During the course of his experiment, the living organism, bacteria, has changed in the physical form. When Streptococcus pneumonia bacteria are grown in the culture plate, some produce the smooth, shiny colonies, while others produce the rough colonies. This is because the S strain bacteria have a mucus polysaccharides coat, while the R strain does not. Mice infected with the S strain's virulent die from pneumonia infection, but the mice infected with R strain do not develop pneumonia. S strain inject to the mice, mice die. R strain inject to the mice, mice live. Griffith was able to kill the bacteria by heating them. He observed that heat-killed S strain bacteria injected into the mice did not kill them. When he injected the mixture of the heat-killed S and R live bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transferred by the heat-killed S strain bacteria. Some transforming principle transferred from the heat-killed S strain had enables to the R strain to synthesize the smooth polysaccharides coat and become virulent. This must be due to the transfer of the genetic material. However, the biochemical nature of the genetic material was not defined from this experiment. Biochemical characteristics of the transforming principles. Prior at the work of the Oswald Avery, Cloyne MacLeod and the Melvin McCarthy in 1933 to 44, the genetic material was thought to be the protein. They worked to determine the biochemical nature of the transforming principle in the Griffith experiment. They purified biochemical proteins, DNA, RNA, etc. from the heat-killed S cell to see the one could transform live R cell into the S cell. They discovered that DNA alone from the S bacteria are caused R bacteria to become transformed. They also discovered that the protein digesting enzyme proteases and the RNA digesting enzyme RNases did not affect the transformation. So, to the transforming substance was not a protein of the RNA. Digestion with the DNAs did inhibit transformation. Suggestion for the DNA caused the transformation. They concluded that DNA is hereditary material, but not all biologists were conceived. Can you think of any differences between DNA and RNA? The genetic material is DNA. The unequivocal proof of the DNA is the genetic material came from the experiment of the Alfred Hershey and the Martha Chase in 1952. They worked with the virus that affect bacteria, also bacteriophages. The bacteriophages also to be the bacteria and its genetic material then enters to the bacterial cell. The bacterial cell treats the viral genetic materials as if they was its own and subsequently manufactures more viruses particle. Hershey and Chase worked to discover whether it was protein or DNA from the virus and entered the bacteria. They grew some viruses on the medium that contained radioactive phosphorus and some of others of the medium contained radioactive sulfur. Viruses grown in the presence of the radioactive phosphorus combine radioactive DNA but not radioactive protein because DNA contains the phosphorus but the protein does not. Therefore, the viruses grown in the radioactive sulfur contain radioactive protein but not a radioactive DNA because DNA does not contain sulfur. Radioactive phages are allowed to attach the E. coli bacteria, then it is the infection proceeds. The viral coat are removed from the bacteria by agitating them in the blender. The viruses practicals were separated from the bacteria by spinning them into the centrifuge. Bacteria which was infected through the viruses that had radioactive DNA, while the viruses to the bacteria does not react infected with the viruses that had the radioactive protein with not radioactive. That indicates that the protein did not material have a passed from virus to the bacteria. Properties of genetic material DNA versus RNA. From the foregoing discussion, it is clear that the debate between the proteins and viruses of DNA as a genetic material was equationally resolved, and the Hershey-Chase experiment is as an established fact that is the DNA fact also a genetic material. However, it is subsequently become clear that in some viruses, RNA as a genetic material, for example, tobacco mosaic virus, QB virus. Answer to the some questions such as why the DNA is the predominant genetic material whereas RNA perform the dynamic functions. of the messenger and receptor has to be found from the differences between the chemical structure of the two nucleic acid molecules. Can you recall the two chemical differences between the DNA and RNA? A molecule that can act as a genetic material must be fulfill the following criteria. It should be able to generate its replication. It should be chemically and structurally be stable. It should provide the scope for the slow changes, mutation that are required for evolution. It should be able to express itself in the form of Mendelian characteristics. If one examine each requirement one by one because of the rule of the base pairing and the complementary, both the nucleic acid DNA and RNA have ability to direct their duplications. The other molecules in the living system such as proteins fail to fulfill first criteria itself. The genetic material should be stable enough not to change with the different stages of life cycle, age or with the changes in the physiology of the organism. Stability as one of the properties of the genetic material was very evident in the Griffith's transforming principle. Itself that heat, which killed the bacteria, at least did not destroy some of the properties of the genetic material. This now can easily be explained in the light of the DNA. That is, the two strands being complementary be separated by heating come together. When appropriate conditions are provided, further two OH groups are present at every nucleotide in RNA is the reactive group and makes the RNA labile and easily degradable. RNA is also now known as the catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acid, the DNA is better genetic material. In fact, of the presence of the thymine at the place of the uracil also confers additionally stability to the DNA. Both the DNA and RNA are able to mutant, in fact of the RNA being unstable mutant at the faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutant and evolve faster. RNA can directly code for the synthesis of protein, hence can be easily expresses the characters. DNA, however, is dependent on the RNA for synthesis of protein. The protein synthesizing machinery has evolved around the RNA, and above discussion indicates that both DNA and RNA as a function as genetic material. But DNA being more stable is preferred for shortage of the genetic information. For the transmission of genetic information, RNA is better. RNA world. From foregoing discussion, an immediate question becomes evident: which is the first genetic material? It shall be discussed in detail in the chapter of chemical evolution. But briefly, we shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest the essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to acts as a genetic material as well as the catalyst. There are some important biochemical reactions in the living system that are catalyzed by RNA catalysis and not by protein enzyme. But RNA being the catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA being double-stranded and having complementary strands further resist changes by evolving the process of the repair. Replication. While proposing the double helical structure of the DNA, Watson and Crick had immediately proposed a scheme for replication of DNA to quite their original statement, that is as follows. It has not except more of notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, 1953. The scheme suggested that the two strands would be separated and acts as a template for the synthesis for new complementary strands. After the completions for replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as a semi-conservative DNA replication. The experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown first in the Escherichia coli and subsequently in the higher organisms such as plants and human cells. Matthew Meslon and Franklin Sahele performed the following experiment in 1958. They grew E. coli in a medium containing NH415 Cl as only the nitrogen source for many generations. The result was the N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. These heavy DNA molecules could be distinguished from the normal DNA by centrifugation is a cesium chloride CSCl density gradient. They transferred the cells into the medium with normal NH4Cl14 and took the samples at various definite time intervals as the cells multiplied and extracted the DNA that remained a double-stranded helical. The various samples were separated independently on CS gradient to measure the densities of DNA. Can you recall what centrifugal force is and think why a molecule with the higher mass density would sediment faster? Thus, the DNA that was extracted from the culture one generation after the transfer of N15 to N14 medium, that is, after 20 minutes, E. coli divides in 20 minutes, had a hybrid or intermediate densities. DNA extracted from the culture after the another generation, that is after 40 minutes, second generation was composed of equal amount of these hybrid DNA and of light DNA. E. coli was allowed to grow for 80 minutes. Then what would be the proportion of light and hybrid densities DNA molecule? Very similar experiment involving use of the radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes was performed on the Vassia faba by Tyler and the colleagues in 1958. The experiment proved that the DNA in chromosomes also replicates semi-conservatively. The machinery and the enzymes. In the living cells such as E. coli, the process of replication requires a set of the catalyzed. The main enzyme is referred to the DNA-dependent DNA polymerase. Since it uses the DNA template to catalyze the polymerization anti-deoxynucleotides, these enzymes are highly efficient enzyme as they have to catalyze the polymerization of a large number of the nucleotides in the very short period of time. E. coli that has only 4.6 into 10 to the power 6 base pair, it with human whose diploid content is 6.6 into 10 to the power 9 base pair, completes the process of replication within 18 minutes. That means the average rate of the polymerization has to be approximately 2000 base pair per second. Not only do these polymerize have to be the fast, but they also have to catalyze the reactions with high degree of accuracy. Any mistake during the replication will result in the mutation. Furthermore, energetically, replication is a very expensive process. Deoxyribonucleosides triphosphatase serves dual purpose in addition to acting the substrate. They provide energy for polymerization reaction. The two terminal phosphates in the deoxynucleoside triphosphatase are high energy phosphate, same as in case of ATP. In addition to DNA-dependent DNA polymerase, many additional enzymes is required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of the DNA separated in its entire length, due to the very high energy requirement, the replication occurs within the small opening of the DNA helix referred to the replication fork. The DNA-dependent DNA polymerase catalyzes polymerization only the one direction, that is 5' to 3'. This creates some additional complications at the replicating fork. Consequently, on one strand, the template with polarity 3' to 5', the replication is continuous, while on the other, the template with the polarity 5' to 3', it is the discontinuous. The discontinuously synthesized fragment are later joined by the enzyme DNA ligase. The DNA polymerase on the other own cannot be initiate the process of replication. Also, the replicate does not initiate randomly at any place of the DNA. There is a definite region in oculi DNA where the replication originates. Such region is termed as a origin of replication. It is because of the requirement of the origin of the replication that a piece of the DNA is, if needed, to be proposed during the recombinant DNA producers required a vector. These vectors provide the origin of replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of the DNA takes place at S shape of the cell cycle. The replication of DNA and the cell division cycle should be highly replicated result into the polyploidy. You will learn the detailed nature of the origin and the processes occurring at this site in higher classes. Transcription. The process of copying genetic information from one strand to the DNA into RNA is termed as a transcription. Here also the principle of the complementary governs the process of transcription except the adenosine now form base pair with the uracil instead of thymine. However, unlike the process of replication, which one set in the total DNA of an organism gets duplicated. In transcription, only a segment of DNA and only one of the strands is copied into the RNA. These necessities defining the boundaries that would be demarcate the region and the strands of DNA that would be transcribed. Why both the strands are not copied during transcription has the simple answer. First, if both strands act as a template, they would code for RNA molecule with different sequences. Remembering complementary does not mean identical. And in turn, they code for proteins. The sequence of amino acid in the proteins would be different. Hence, one segment of the DNA would be coding for the two different protein, and this would complicated to genetic information transferred mechanically, machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent the RNA form being untranslated into the protein and the exercise of the transcription would become a futile one. Transcription unit. A transcription unit in the DNA definitely primary in by three regions of the DNA, a promoter, a structural gene, and a terminator. There is a conservation of the definiting of two strands of the DNA in the structural gene of the transcription unit. Since these two strands have opposite polarity and the DNA-dependent RNA polymerase, they also categorize the polymerization in only one direction, that is 5' to 3', and the strands that has polarity 3' to 5' acts as a template, and it is referred to as the template strand. The other strand which has the polarity 5' to 3' and the sequences of the same as a RNA. It is displaced during the transcription. Strategy that this strand is referred to coding strand. All the references point while defining transcription until it made with the coding strands. To explain the point of the hypothetical sequence from the transcription unit is represented below: template strand and coding strand. The promoter and the terminator flank of the structural gene in a transcription unit. The promoter is said to be located towards the 5' end upstream of the structural gene and referred to made with the respect to the polarity of the coding strand. It is the DNA sequence that provides binding site for RNA polymerase, and it is the presence of promoter in the transcription unit that also defines the template and coding strands by switching its position with terminator. The definitions for coding and template strands could be reserved. The terminator is located towards the 3' end downstream of the coding strand that is usually defines the end of the process of transcription. There are additional regularity sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with the regulations of gene expression. Transcription unit and the gene. A gene is defined as the functional unit of inheritance. Through there is no ambiguity that is the genes were located on the DNA. It is difficulty to literally define a gene in terms of the DNA sequence. The DNA sequence coding for the tRNA or rRNA molecule also defined a gene. However, by defining the cistron as a segment of DNA coding for the polypeptide, the structural gene in the transcription unit could be said as a monocistronic or polycistronic. In eukaryotes, the monocistronic structural gene have interrupted the coding sequence and the genes in eukaryotes are split. The coding sequences were expressed as a sequence as defined as exons. Exons are said to be the sequence that appears in the mature of the process of RNA. The exons are interrupted by the introns. Introns intervening the sequence do not appear to the mature or processed RNA. The split gene arrangement further complicates the definition of the gene in terms of the DNA segment. Inheritance of the characters is also affected by the promoter and regulatory sequences of the structural gene. Hence, sometimes the regulatory sequence are loosely defining the regulatory genes, even though these sequences do not code for any RNA or protein. Types of the RNA and the process of the transcription. In bacteria, there are three major types of the RNAs: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. Then mRNA provides the template. tRNA brings amino acid and reads the genetic code. And rRNA plays structural or catalytic role during translation. There is a single DNA-dependent RNA polymerase that is catalyzed transcription of all types of the RNA in bacteria. RNA polymerase binds to the promoter and initiate initiation. It is used nucleoside triphosphatase as a substrate and polymerize in a template-dependent fashion following a rule of complementarity. It somehow also facilitates opening of the helix and continuous elongation. Only a short stretch of the RNA remains bound to the enzyme. Once the polymerase reaches and terminator region, the nascent RNA falls off and also the RNA polymerase. This results in the terminations of the transcription. In interrogating questions is that how is the RNA polymerase able to catalyze all the three steps, which are infections, elongation and termination. The RNA polymerase is only capable of the catalyzing the process of elongation. It associated transcriptally with initiation factors respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. In bacteria, since the mRNA does not require any processing to become active, and also since the transcription and translation takes place in the same compartment, there is no separation of the cystole and nucleus in bacteria. Many times the translation can be begun much before the mRNA is fully transcribed subsequently. Consequently, the transcription and the translation can be coupled in bacteria. In eukaryote, there are two additional complexities. There are at least three RNA polymerase in the nucleus in addition to the RNA polymerase found in the organelles. There is a clear-cut division of the labor. The RNA polymerase 1 transcribed rRNAs 28S, 18S, 5.8S, whereas the RNA polymerase 3 is responsible for transcription of tRNA, 5SR RNA and snRNAs, small nuclear RNAs. The RNA polymerase 2 transcribed the precursor of the mRNA and heterogeneous nuclear RNA, hnRNA. The second complexity is that the primary transcribed contain both the exon and introns and are non-functional. Hence, it is an exons are joined in the definite order. hnRNA undergoes additional processing is called capping and tailing. In capping, the unusual nucleotide guanosine triphosphate is added to the 5' end of the hnRNA. In tailing, the adenosine residues are added in the 3' end of the RNA. Template independent manner. It is fully added in 3' end processed in hnRNA, now called mRNA, that is transported out of the process for translation. The significance of such a complexity is now bringing to be the understand and the split gene arrangement respect probably ammonium. The significance of the such complexity is now bringing to be the understood and split gene arrangement represent probably an ancient further of the genome. The presence of the intron is thus remains of the antiquality as the process of the splicing regenerate represents the dominance of the RNA world. In the recent time, the understanding of RNA and RNA dependent processes in the living system have assumed more importance. Genetic code. During replication and transcription, the nucleic acid was copied to form the another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires the transfer of genetic information from the polymer of the nucleotide to the polymer of the amino acid. Neither does any complementarity exist between the nucleotides and amino acid, nor could be any down on the theoretically. These excited ample evidence do to support the notice that changes in nucleic acid were responsible for change into amino acid in protein. These lead to the proportion of the genetic code that would direct the sequence of the amino acid during the synthesis of protein. If determining the biochemical nature of the genetic material and the structure of the DNA was very exciting, the proportion of the differing of genetic code were not challenging. In very true sense, it requires involvement of the scientists from several disciplines, phycologists, organic chemistry, biochemists, and genetics. It was the George Gamow and the physicists who argued that the science had are many four bases, and if they have a code for 20 amino acid, the code should be constituted combinations of the bases. He suggested that in order to the code for the all 20 amino acids, the code should be made up of 3 nucleotides is a very bold proportion because the prematuration combination is 4 s to power 3 4 into 4 into 4 would generate 64 codons, generating many more codons that requires. Providing the proof that is the codon was triplet was more doubting task. The chemical method developed by the Har Govind Khorana was instrumental in synthesis of rRNA molecules which defined the combinations of bases. Marshall Nigres and the cell system for the protein synthesis finally helped the code for the describing several occasional enzyme. Was also helpful for the polymerization RNA was with the different sequences in the template independent manner. Finally, in checkerboard for the genetic code was prepared which is given. The silent features of the genetic code. The codon is triplet. 61 codons are codes for the amino acid. 3 codons do not code for any amino acid, hence they function as a stop codon. 1 codon codes for only one amino acid, hence it is unambiguous and specific. Some amino acids are coded by more than one codon, hence the code for degenerated. The codon is read by the amino acid is coding continuously fashion. There are no punctuations. The code is nearly universal. For example, from bacteria to the human, UUU would code for the phenylamine. Some exceptions to these rules have been found in mitochondrial codons and some protozoans. AUG has a dual function. It codes for the methionine and it also acts as the initiator codon. If involving in the sequence of the nucleotides in mRNA predict the sequence of the amino acid coded by it. Now try to opposite following its sequence of the amino acid coded by the mRNA predict the nucleotide sequence in the RNA. Met, pe. Do you face any difficulty to predicting the opposite? Can you now correlate which two properties of the genetic code you have learned? Mutation and genetic code. The relationship between the genes and the DNA are best understood by mutation studies. You have studied about the mutation by its effect in chapter 5. Effects of the large deletion and rearrangement of the segment of DNA are easy to compare. It may result in loss of gain of the gene and so and function. The effect of the point mutation will be explained here. A classical example of the point mutation is change the single base pair in the gene for beta globin chain, which result in the change in the amino acid residue glutamine to valine. It results in the disease called as the sickle cell anemia. Effect of the point mutation that insert and deletes the base of the structural gene can be better understood by given following simple example. Consider a statement that is made up of following words, each having the three letters like a genetic code. Ram has red cap. If we insert the letter B between the has and red, the rearrange the statement, it would be read as Ram has DCAP. Similarly, if we now insert the two letters of the same place of the B, I now would be read Ram has BBREDCAP. Now we insert the three letters together, say big. The statement would read Ram has a big red cap. The same exercise can be repeated by deleting the letters R, E and D one by one. The rearranging statement to make the triplet word Ram has EDCAP, Ram has DCAP, Ram has cap. The conclusion from the above exercise is very obvious. Insertion and deletion of one or two bases changes the reading frame from the point insertion or deletion. Insertion or deletion of three of its multiple bases. Insert or delete one or multiple codon, hence one or multiple amino acids are reading frame remains unaltered from the point onwards, such as the mutation is referred to the frame shift, insertion or deletion mutation. This forms the genetic basis for the proof that codon is a triplet. It is read in a continuous manner. tRNA, the adapter molecule. From the very beginning of the proportions of the code, it was clear to Francis Crick that there has to be mechanical to read the code that also link in the amino acid. Because amino acid have no structural specialties to read the code uniquely, they postulated the presence of the adapter molecule that would on the hand read to code, on the other hand would bind to specific amino acid. The tRNA, then it is called the sRNA or soluble RNA, was now before the genetic code was postulated. However, its role is and the adapter molecules has assigned much later. tRNA has an anticodon loop that has bases complementary to the codes, and it is has the amino acid acceptor end which is bind to amino acid. tRNAs are specific for the each amino acid. For initiation, there is the another specific tRNA is referred to be the initiation tRNA. There are no tRNAs as stop codon. The secondary structure of the tRNA has been depicted to look like a clover leaf, and actual structure the tRNA is a compact molecule which looks like a inverted L. Translation refers to the process of polymerization of amino acid to form polypeptide. The order and the sequence of the amino acid are defined by the sequence of the bases in the mRNA. The amino acid are joined by the bond which is known as the peptide bond. Formation of the peptide bond require energy, therefore it is the first phase itself. Amino acid are activated in presence of ATP and linked to their cognate of tRNA. A process is commonly called the charging of the tRNA or amino acetyl of tRNA. To be more specific, if two such charged tRNAs are brought close enough, the formation of the peptide bond between them would be favored energetically. The presence of the catalyst would enhance the rate of the peptide bond formation. The cellular factor responsible for the synthesizing protein is the ribosome. The ribosome consists of the structural RNAs and about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of the translation of the mRNA to protein begins. There are two sites in the large subunit for subsequent amino acid to bind to, thus be close enough to each other for the formation of a peptide bond. The ribosome also acts as a catalyst for the formation of peptide bond. A translational unit of the mRNA is the sequence of the RNA that is flanked by the start codon AUG and the stop codon that codes for the polypeptide. An mRNA also has some additional sequences that are not translated and are referred as an untranslated region UTR. The UTRs are present and are referred as an untranslated region. The untranslated are present at both 5' and 3' They are required for efficient translation process. For initiation, the ribosome binds to the mRNA at the start codon AUG that is recognized only by the initiation of tRNA. The ribosome proceeds to the elongation of the phase of the protein synthesis. During this stage, complexes compound of the amino acid linked to tRNA sequentially bind to the appropriate codon of mRNA by forming complementary base pair with the tRNA anticodon. The ribosome moves from the codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequences detected by DNA and represented by mRNA. At the end of release, factor bind to the top codon, terminating translation and releasing the complete polypeptide from the ribosome. Regulation of gene expression. Regulation of gene expression refers to the very broad term that may occur at various levels, considering that gene expression result in the formation of polypeptide. It can be regulated at several levels. In eukaryotes, the regulation could be exerted at transcriptional level, processing level, transport of mRNA from nucleus to the cytoplasm, and translational level. The gene in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of the disaccharides, lactose into galactose and glucose. The bacteria use them as a source of energy. Hence, if the bacteria do not lactose around them to the utilize for the energy source, they would no longer require for the synthesis of the enzyme beta-galactosidase. Therefore, in simple terms, it is metabolic, physiological, and environmental conditions that regulate the expression of the gene. The development and differentiation of the embryo into adult organism are also a result of the coordination of the regulation of the expression of the several sets of gene. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for the control of the gene expression. In transcriptional unit, the activity of RNA polymerase at a given promoter is in turn regulated by the interaction with accessory protein, which affect its ability to recognize start sites. These regulatory proteins can be act both positively and negatively. The accessibility of the promoter region of the prokaryotic DNA is in many cases regulated by the interaction of the protein with sequences termed operators. The operators region is adjacent to the promoter elements in most of the operands, and in most cases the sequences of the operator binds a repressor protein. Each operand has its specific operator and specific repressor. For example, lac operator is present only in the lac operon, and it interacts specifically with lac repressor only. The lac operon. The elucidation of the lac operon was also a result of the close association between the genetist Francisco Jacob and the biochemist Jacob Monod. They were first these included transcriptionally regulated system in lac operon. A polycystonic structural gene is regulated by the common promoter and regulatory genes. Such arrangement is very common in bacteria and is referred as to operon. To the name few such examples like operon, trap operon, ora operon, his operon, well operon, etc. The lac operon consists of one regulatory gene, that is I gene. Here the term I does not refer to the inducer, rather it is derived from the word inhibitors, and three structural genes Z, Y and A. These I gene codes for the repressor of the lac operon and Z gene codes for the beta galactosidase, which is primarily responsible for the hydrolysis of the disaccharides. Lactose into its monomeric units, galactose and glucose, and Y gene codes for the primase, which increases the permeability of a cell to the beta galactosides. The alpha gene is encodes the transcriptase. Hence, all the three genes produced in a lac operon are required for metabolism of lactose. In most other operons as well, the genes represent in the operon are needed together to function in the same related metabolic pathway. Lactose is the substrate for the enzymes of beta galactosidase and it regulates the switching on and off of the operon. Hence, it is termed as an inducer. In absence of the preferred carbon source, such a glucose, if the lactose is provided in the growth of the medium of the bacteria, the lactose is transported into the cells through the actions of primase. The lactose then induces the operon in the following manner. The repressor of the operon is synthesized all the time constitutively from the I gene. The repressor protein binds to the operator region of the operon and prevents the RNA polymerase forming transcribing the operon. In the presence of the inducer such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, regulation of lac operon can be visualized as a regulation of enzyme synthesis by its substrate. Remember, glucose and galactose cannot act as an inducer for lac operon. Can you think for how long the lac operon would be expressed in presence of the lactose? Regulation of the lac operon by repressor is referred to as negative regulation. Lac operon is under control of the positive regulation as well, but it is beyond the scope of the discussion at this level. Human Genome Project. In the preceding section, you have learned that it is the sequence based on the DNA that determines the genetic information of a given organism. In other words, the genetic makeup of an organism or an individual lies in a DNA sequence. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions lead to the quest of finding out the complete DNA sequence of human genome. With the establishment of the genetic engineering technique where it was possible and isolate and clone any piece of the DNA and availability of a simple and last techniques for the determining DNA sequences. A very ambitious project of the sequencing human genome was launched in year 1990. Human Genome Project HGP was called as a mega project. You can imagine the magnitude of the requirements for the project if we define the aims of the project as follows. Human genome is said to have approximately 3 into 10 to the power 9 base pair, and if the cost of the sequencing requires in $3 per base pair, estimate the cost of the beginning. The total estimate cost of the project would be approximately $9 billion US. Further, if the obtained sequence were to be stored in the typed form in the books, and if each page of the book contained 1000 letters and each book contained 1000 pages, then 3,300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of the data expected to be generated also necessitates to use a high speed of the consumptional device for the data storing and retrieval and analysis. HGP was closely associated with the rapid development of the new area in the biology called bioinformatics. Goals of HGP. Some of the most important goals of the HGP were follow. Identify all the approximately 20,000 to 25,000 genes in the human DNA. Determine the sequence of the 3 billion chemical base pairs that make up of human DNA. Store this information in databases. Improve tools for the data analysis. Transfer related technologies to other sectors such as industries, addresses the ethical, legal, and social issue (LSI) that may arise from the project. The Human Genome Project was a 13-year-old project coordinated by the US Department of the Energy and the National Institute of Health. During the early years of the HGP, the Wellcome Trust UK became a major partner. Additional contribution came from Japan, France, Germany, China, and others. The project was completed in 2003. Knowledge about the effect of the DNA variations among the individuals can be lead to revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect the human being. Beside providing clues to the understanding human biology, learning about non-human organisms, DNA sequencing can lead to an understanding too of their natural capabilities that can be applied towards the solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non-human model organisms such as bacteria, yeast, Caniobacterium elegans, Drosophila plants have also been sequenced. Methodologies. The method involved in two major approaches. One's approach focused on identifying all the genes that are expressed as RNA. The other took the blind approach to simplify sequencing the whole set of the genome that is combine all the coding and non-coding sequences. The latter assigning different region in the sequence with function. For sequencing, the total DNA from a cell is isolated and converted into random fragment of relatively smaller size. Recall DNA is a very long polymer and there are technical limitations in the sequencing of very long piece of the DNA. And the clone is suitable host using the specialized vector and cloning resulted in the amplification of each piece of DNA fragment so that it subsequently could be sequenced with yields. The commonly used hosts were bacteria and yeast, and the vectors were called the BAC, bacterial artificial chromosome, and YAC, yeast artificial chromosome. The fragments were sequenced using automated DNA sequencers that worked on the principles of a method developed by Frederick Sanger. Remember, Sanger is also credited for developing method for the determination of amino acid sequences in protein. These sequences were then arranged based on the some overlapping region present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequences of the chromosome 1 was completed only in May 2006. This was the last of the 24th human chromosome, 22 autosome and XY to be the sex chromosome. Another challenging task for assigning the genetic and the physical map of the genome, this was generated using the information of the polymorphism of restriction endonuclease recognized sites and some repetitive DNA sequence known as the microsatellite. One of the applications of the polymorphism in the repetitive DNA sequence shall be explained in the next section of the DNA fingerprinting. Silent feature of the human genome. Some of the silent observations drawn from the Human Genome Project are as follows. The human genome contains 164.7 billion nucleotide bases. The average gene consists of 3000 bases but size vary greatly, with the largest known human gene being dystrophin in 2.4 billion bases. The total number of the gene is estimated 30,000, much lower than the previous estimate 80,000 to 1,40,000 genes. Almost all the nucleotide bases are exactly in the same in all people. The functions are unknown for over 50% of the discovered gene. Less than 2% of the genome codes for protein. Repetitive sequences make up a very large portion of the human genome. Repetitive sequences are stretched of the DNA sequence that are repeated many times, and sometimes 100 to 1000 times. They are thought to have no direct coding function, but they shed light on the chromosome structure, dynamics, and evolution. Chromosome 1 has the most gene, 2968, and the Y has the fewest, 231. Scientists have identified about 1.4 billion locations where single base of DNA differences SNPs occur in humans. This information promises to revolutionary process by finding the chromosomal location for the disease associated sequence and tracing the human history. Application and future challenges. Deriving meaningful knowledge from the DNA sequencing will define research through the coming decades, leading to our understanding of the biological system. This enormous task will require the expertise of the creativity of the tens thousands of the scientists from the varied disciplines in both the public and the private sector worldwide. One of the greatest impact of the having of the HG sequencing may will be enabling a radically new approaches to biological researches. In the past, researchers study one or few genes at a time. With the whole genome sequence and new high through throughputs technologies, we can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcript in the particular issue or organ or tumor, or how tens of the thousands of the gene and proteins work together in interconnected networks to the outrage of the chemistry of life. DNA fingerprinting. As stated in the predicing section, 99.9% of the base sequencing among the humans is same. Assuming the human genome 3 into 10 to the power 9 base pair, in how many base sequences would there be differences? It is these differences in the sequence of the DNA which makes every individual unique in their phenotypic appearance. If one aimed to find out genetic differences between the two individuals or among the individuals of a population, sequencing the DNA every time would be daunting and expensive task. Imagine trying to compare the two sets of the 3 into 10 to the power 6 base pairs of DNA. Fingerprinting is very quick way to compare the DNA sequences of any two individuals. DNA fingerprinting involves the identifying different of same species region in the DNA sequence called as a repetitive DNA. Because in this sequence, a small sketch of the DNA is repeated many times, these repetitive DNA are separated from the bulk genome DNA as different peak during the density gradient centrifugation. The bulk DNA formed a major peak and the other small peak are referred to as the satellite DNA. Depending on the base composition, A S to T reach, G S to C reach, length of segment and number of the repetitive units. The satellite DNA is classified into many categories such as microsatellites, mini satellites, etc. These sequences normally do not code for many proteins, but they form a large portion of human genome. These sequences shows a high degree of the polymorphism and form the basis of the DNA fingerprinting. Since DNA from every tissue such as blood, hair, follicles, skin, bone, saliva, sperm, etc. form an individual show a same degree of the polymorphism, they become very useful identification tool for the forensic application. Further, as a polymorphism are inheritable from parents to children. DNA fingerprinting is the basis for patterning testing in the case of disputes. As a polymorphism in DNA sequence is the basis of the genetic mapping of human genome as well as the DNA fingerprinting. It is essential that we understand what DNA polymorphism means in simple terms. Polymorphism variation in the genetic level arises due to the mutation. Recall different kind of the mutation and their effect that you have already studied in chapter 5 and in the preceding section in this chapter. New mutation may arise in individuals either in the somatic cells or in the germ cells. The cells that generate gametes in sexually reproducing. If a germ cell mutation does not seriously impair individuals' ability to have an offspring who can transmit the mutation, it can spread to the other member of the population through the sexual reproduction. Allelic, again recall the definitions of the allele of chapter 5. Sequencing variation has a traditional being described as a DNA polymorphism if more than one variant allele at a focus locus occurs in the human population with the frequency of greater than 0.01. In simple terms, if the inheritable mutation is observed in the population at high frequency, it is referred to as a DNA polymorphism. In probability of such a variations to be observed in non-coding DNA sequence would be higher at mutation in these sequences may not have any immediate impact in an individual reproductive ability. These mutations keep on accumulating generations after generation and form one of the bases of the variability polymorphism. These there is a variety of different types of the polymorphism ranging from the single nucleotide change to very large scale changes. For evolution and the speciation, such a polymorphism play a very important role and you will study these in details in higher classes. The techniques of the DNA fingerprinting was initially developed by Alec Jeffrey. He used a satellite DNA as a probe that shows a very high degree of polymorphism. It was called as a variable numbers of the tendon repeats. The technique as earlier involved southern blot hybridization using the radio-labeled VNTR as a probe include isolation of DNA, digestion of the DNA by restriction endonuclease, separation of the DNA fragments by the electrophoresis, transferring of the separated DNA fragment to the synthetic membrane such as nitrocellulose or nylon, hybridization using the labeled VNTR probe, and detection of the hybrids of the DNA fragment by autoradiography. A systematic representation of the DNA fingerprinting has shown. The VNTR belongs to the class of satellite DNA referred to be the mini satellite. A small DNA sequence is arranged randomly in many copy numbers and the copy of the numbers varies from the chromosome to chromosome in an individual. The number of the repeat shows very high degree of the polymorphism. As a result, the size of VNTR varies in size from 0.1 to the 20 kb. Consequently, after hybridization of the VNTR probe, the autoradiogram gives many bands of the differing size. These bands give a characteristic pattern for the individual's DNA. It differs from the individuals to individuals in the population except in case of the monozygotic twins. The sensitivity of the technique has been increased by the use of polymerase chain reaction PCR. You will study about it in the chapter 11. Consequently, DNA from the single cell is enough to perform DNA fingerprinting analysis in addition to the application of forensic science. It has much wider applications such as the determining population and genetic diversibilities. Currently, many different probes are used to generate DNA fingerprints.